Welcome to this explainer. Okay, today we're putting on our lab coats and playing Reef Detective to uncover the hidden, microscopic assassins that are actually responsible for killing your corals. Because let's be honest, if you've ever watched a prized coral wither away while the rest of your tank absolutely flourishes, you know exactly how frustrating and wildly expensive this hobby can be. Well, we're going to get to the bottom of it today.
So when a coral starts inexplicably declining in your tank, what's literally the first thing we all do? We immediately blame broad environmental factors, right? We frantically test the alkalinity, we check for temperature swings, or we just start pointing fingers at overall water quality. But the truth is often way stealthier than a simple chemistry imbalance.
To actually solve this, we need a massive paradigm shift. We have to stop looking exclusively at the broad threats affecting the whole tank environment, the temperature swings and water quality issues, and start actively hunting for host-specific threats. These are highly specialized pests that selectively attack particular corals. They zero in on a single host, turning what looks like random bad luck into a really persistent, localized, colony-level problem. Suspect number 1. The tissue eaters.
Let's talk about Montipora eating nudibranchs. These are highly adapted sea slugs that specifically zero in on Montipora tissues. They are absolute masters of camouflage, often taking on the exact color of the tissue they just consumed. This makes them perfectly disguised during your routine reef checks. And because they're so tiny and brilliantly hidden, early signs of their presence are almost always mistaken for minor environmental stress, at least until the tissue loss is completely undeniable.
Now these guys leave behind a very specific fingerprint of damage. You really want to watch out for paling tissue, areas that literally look like they're peeling off, irregular receding edges, and ultimately stark white exposed skeletal patches. And keep in mind, this situation spirals out of control incredibly fast if you have multiple individuals breeding, especially if you're casually moving Montipora fragments between tanks without doing a thorough inspection first.
Moving on to suspect number 2, the crevice hiders.
Now, I definitely want to be clear here, not all crabs hanging out in your Acropora are bad guys. Some are actually totally harmless or even beneficial commensals. But the harmful species, they are absolute masters of evasion. They feed right on the coral tissue or actively disturb the polyps, and the second you come looking for them, they retreat deep down into the intricate crevices or right at the base of the colony structure just to avoid removal.
Unlike generalized tank stress that kind of just pales a whole coral uniformly, a crab's signature damage is super patchy. You're going to see localized paling and missing polyps in highly specific spots across the branches. Most importantly, though, the damage relentlessly tracks the exact same coral over time. If you suspect these crabs, your absolute best bet is careful observation during feeding times and targeted, close-up inspections down at the colony's base.
And finally, suspect number 3, the microparasites. Parasitic copepods really take stealth to an entirely new level. We're talking about microscopic crustaceans that attach themselves directly to the coral tissue. Because they are nearly impossible to detect with the naked eye, these little guys contribute to an ongoing, long-term stress that looks incredibly subtle at first. You won't see them scurrying away like the crabs. You'll just see a coral that simply refuses to thrive.
And this is exactly why caretakers get so incredibly frustrated. The symptoms of copepods perfectly mimic other stressors, like nutrient imbalances or low-flow irritation. Hobbyists end up constantly chasing phantom water chemistry issues. They tweak their dosers, they change their flow rates, they do massive water changes, all while the real clue is persistence. If one specific coral continuously deteriorates while the surrounding colonies remain totally stable and healthy, you don't have a water chemistry issue. You have a specialist parasite.
The good news? You don't actually need a high-powered microscope to crack this case. You just need a little patience. Watch for persistent symptoms like reduced polyp extension, severely slowed growth, localized irritation, and an eventual visible decline in tissue quality. It's definitely a slow burn, but recognizing these signs on an isolated colony is your critical first step to confirming a microparasite invasion.
Okay, let's put this all together with the diagnostic matrix. Think of this as your ultimate detective's cheat sheet. By cross-referencing the target coral with the specific damage pattern, you can figure out exactly what you're dealing with. If it's a Montipora with peeling tissue and exposed skeletal patches, suspect the nudibranchs. If it's an Acropora with patchy, localized missing polyps, look deep in the crevices for crabs. And if a single, random coral shows slowed growth and reduced extension while its neighbors are totally fine, start thinking copepods. Matching the unique damage pattern to the specific host vulnerability is going to save you precious time in your diagnosis.
Now for the most important part, The counteroffense.
Here is absolutely the most critical takeaway for treatment. An effective response has to start with early detection and a coral-by-coral assessment. We really have to stop treating coral pests as one giant, mysterious category. You can't just dump a broad-spectrum chemical into your tank and hope for the best. Instead, you need to methodically verify the issues by carefully inspecting individual colonies based on their known vulnerabilities.
To actually win this war, we need to look at scientific monitoring strategies. Here's your action plan. Step 1, and honestly, arguably the most vital step, quarantine new coral fragments, prevent the introduction in the first place. Step 2, conduct visual and magnified checks of any suspect colonies. Step 3, carefully remove any visible pests you can find. And Step 4, reduce the environmental conditions that allow for fast population growth. By meticulously following this exact protocol, you can intervene early and preserve your colonies way before the tissue damage becomes irreversible.
I want to leave you with one final, vital question to ponder for your own reef keeping. Ask yourself, are you treating the whole ocean or just the invisible invaders? Stop making panicky, broad adjustments to your water chemistry every single time one coral looks unhappy. Instead, grab a magnifying glass, check your matrix, and start looking closely for the predictable, host-specific problems hiding right in front of you. Until next time, keep observing, keep questioning, and happy reefing.